Hello friends, today's topic of discussion is metabolism of VLDL and formation of LDL. This is an important topic as per new revised MCI curriculum. It can be a part of LAQ long answer question in your university examination. In this topic, we will discuss about the synthesis of VLDL, then various steps in the formation of LDL, then mechanism of LDL uptake, and in short discussion of various hyperlipoproteinemias. So you know the function of VLDL and LDL. VLDL is responsible for transport of endogenous TG, endogenous triacylglycerol, means triacylglycerol synthesized in our body. It is transported from liver to the peripheral tissues, liver to peripheral tissues with the help of this VLDL. And LDL, it is responsible for transport of cholesterol from liver to the extrahepatic tissues or peripheral tissues. LDL, low density lipoprotein, and VLDL means very low density lipoproteins. In the synthesis of VLDL, the synthesis occurs in the liver. The triacylglycerol, cholesterol and cholesterol esters, and phospholipid with ApoB100 forms the VLDL, that is newly synthesized VLDL by the liver, that is known as nascent VLDL. Nascent means newly synthesized. So this is nascent VLDL, very low density lipoprotein. Lipoprotein means complex of various lipids with protein to increase the solubility of this compound and for the transport of these various lipids in the plasma. Now, second step in the VLDL formation or in the metabolism of VLDL is maturation. So, maturation of VLDL occurs by acquiring ApoC2 and ApoE, means apolipoprotein C2 and ApoE from the HDL. Okay, so it acquires these two apolipoproteins from the HDL and converted into mature VLDL. It is mature VLDL. Now this mature VLDL, with the help of lipoprotein lipase, this mature VLDL in the peripheral tissues, there is lipoprotein lipase which is present on the capillary walls. So this lipoprotein lipase, it is activated by ApoC2, which is present in the capillary, on the surface of capillary walls. It causes the conversion of this triacylglycerol into free fatty acid and glycerol. This glycerol is taken up by the liver, while this free fatty acid, it is utilized in the muscle and adipose tissue, mainly adipose tissues. It is peripheral tissues. So this free fatty acid, it is utilized in the peripheral tissues. Now this is converted into chylomicron remnant, which contain less amount of triacyl glycerol as compared to the mature VLDL. So triacyl glycerol, cholesterol and cholesterol ester, it loses ApoC2, there is ApoE and B100. So this is known as VLDL remnant. VLDL remnant or intermediate density lipoprotein. So this is a third step in the VLDL metabolism. The first step is formation of nascent VLDL, means newly synthesized VLDL with incorporation of various lipids with ApoB100. This is nascent VLDL. Then in maturation, it acquires ApoC2 and ApoE from the HDL and it is converted into mature VLDL. Now, by the action of lipoprotein lipase, which is activated by ApoC2, there is degradation of this triacyl glycerol into diacyl glycerol, monoacyl glycerol, and finally free fatty acid and glycerol. This free fatty acid, they are utilized by the peripheral tissues, and glycerol, it is taken up by the liver for further metabolism. And by giving back this ApoC2 to HDL, it is converted into VLDL remnant. And VLDL remnant is also known as intermediate density lipoprotein. So this is intermediate density lipoprotein. Now the fourth step in the metabolism is formation of LDL. This ideal means intermediate density lipoprotein, it has two fates. The first one is either it is taken up by the liver by the receptor mediated endocytosis, and second it is converted into LDL. So it is taken up by the liver by receptor mediated endocytosis, and second is it is converted into LDL. So this is the structure. LDL, it is it mainly contains cholesterol and only one apoprotein, that is apolipoprotein B100. So this is a LDL. The cholesterol, it is from the VLDL. Then B100, apolipoprotein B100, it is the only lipoprotein which is present in the LDL. Got it? It is formed from the intermediate density lipoprotein. Okay. Now the difference is here it contains cholesterol. Now difference in the lipid content of these three means VLDL, ideal, suppose 3, VLDL, ideal and LDL. So VLDL contain near about 60% Tg, VLDL, and it contain 20% cholesterol, 60% Tg, 20% cholesterol, 5% proteins and 15% phospholipids. In intermediate density lipoprotein, the TAG content is 30% and cholesterol content is 30%, and remaining 20% is protein and phospholipids. While in LDL, the cholesterol content is 50%, this triacylglycerol is 10% and 20% phospholipid and proteins. So this is the difference in the content of TAG. It contains 60%, then intermediate density lipoprotein contain 30% and LDL contain 10% of TAG, and the cholesterol content is increased in the LDL. Now LDL, it is taken up by the peripheral tissues. It is taken up by the peripheral tissues and liver. It is metabolized mainly in the liver, in adipose tissue and adrenal medulla. It is adrenal gland, adipose tissue and liver mainly metabolized in these tissues. Now coming to the uptake of LDL. In the uptake of LDL, it is specifically with the help of one protein that is known as clathrin, and the process is known as receptor mediated endocytosis. This is LDL, and LDL receptor, suppose this is membrane, this is plasma membrane, it is a lipid bilayer. Then LDL receptor, it is made up of outer part, then transmembrane protein and inner part. Okay, this is LDL receptor. It is made up of outer protein, transmembrane protein and inner core. Okay, this outer membrane, it is a site for attachment of LDL. This is the LDL. It is the site for attachment of LDL. And this inner core, it is attached with the clathrin. Clathrin, these are the specialized protein which helps in the endocytosis of LDL particles. Now how the endocytosis occurs? So these are the coated pits, these are the pits, and these are clathrin coated pits. On these clathrin coated pits there are receptors. These are the receptors. Now suppose this is a LDL particle. So LDL bound with this receptor with the outer part. So there is formation of LDL and LDL receptors. There is formation of LDL and LDL receptor complex. So this is LDL and this is LDL receptor, and this one is the clathrin. Okay, so it is important MCQ here. Now with the help of this clathrin, the internalization of these coated pits occurs. So there is internalization of these pits and these LDL particles internalized with the help of these LDL receptors. These are the clathrin. Now shading of this clathrin occurs and the receptor part, it is reutilized. So receptor part, it is reutilized. Shading of clathrin occurs. This structure, it is known as endosomes. Endosomes. Okay. Now shading of clathrin occurs and this receptor part reutilized for the further LDL particles, and now it is endosomes with LDL particles which fuses with the lysosomes, primary lysosomes, to form a phagolysosomes. Okay, it is endosomes, endosome fuses with the lysosome to form a endolysosomes. So there is formation of endolysosomes. Okay, now this endolysosome, it contain LDL. It causes the degradation of cholesterol and cholesterol ester which is present inside the vesicle. Okay, so it has three fate. The cholesterol which is present in the endolysosome, or it is in the tissue, it has three fate. First one is the storage. There is re-esterification of cholesterol occurs in the tissue and it gets stored. This reesterification occurs with the help of acyl cholesterol acyl transferase. Acyl CoA cholesterol acyl transferase, it causes reesterification of cholesterol present in here in the tissue and it causes the storage. Further, it is utilized for the plasma membrane synthesis. Then it is utilized for the synthesis of steroid hormones and it is utilized for the synthesis of bile acids. Okay. It also, this cholesterol also decreases the synthesis of LDL receptors and also inhibit the cholesterol synthesis by the tissue. Cholesterol synthesis. So this is the cholesterol in peripheral tissues. It is mainly taken up by the adrenal gland, adipose tissue and mainly by the liver. So it is metabolized in the liver. It is absorbed with the help of LDL receptors which requires ApoB100 for its uptake. Okay. So it is a regulatory process. It is a regulatory process. Increase in the LDL level leads to the formation of oxidized LDL and glycated LDL. This is very, very important. So oxidized LDL, increased production of oxidized LDL causes its uptake by the macrophages. So it is a scavenger receptors which is present in the macrophages which causes the absorption or uptake of this LDL particles, and when it engage with these macrophages or engage with the LDL or when it engage with the cholesterol, they leads to the formation of foam cells. And these foam cells are responsible for formation of plaques, which is responsible for atherosclerosis. That's why in most of the book it is mentioned that LDL is a bad cholesterol because it increases the atherosclerosis and coronary artery diseases. Okay, so this is the receptor mediated endocytosis of LDL particles. Okay, now with these three lipoproteins, that is VLDL, IDL and LDL, there are three disorders which are related with these three lipoproteins. The first, VLDL, it is related with type 4 hyperlipoproteinemia. It is also known as hypertriglyceridemia. Okay, type 4 hyperlipoproteinemia. It is related with the VLDL metabolism. Then IDL, it is related with, that is dis beta lipoproteinemia or type 3 hyperlipoproteinemia or also known as dis beta lipoproteinemia. And the LDL, it is related with type 2, that is LDL receptor defect in LDL receptor. So these three hyperlipoproteinemias, they are related with these lipoproteins. Summarizing all, there are 4 important steps in the VLDL metabolism. First one is the synthesis of nascent VLDL. Second one is the maturation. Third one is the formation of IDL or formation of VLDL remnant. And fourth one is the formation of LDL from the IDL. The TG content, it decreases with this formation while the cholesterol content increases from VLDL to IDL to LDL. And this LDL, it is mainly taken up by the peripheral tissues, which contain adrenal medulla, adipose tissue, adipose and adrenal gland. Okay, and the liver, it causes the uptake of this LDL particle. So this is all about LDL. Keep watching. Thank you.